In this video, we will be studying the classification of soil organisms. If you have watched the previous video, you may have idea about the soil biology, what is the definition of it, and history related to the soil biology, and also the important scientists and their contribution, because these are some topics that can be asked in various competitive exams. So in this video, we will be focused on the classification of soil organisms. So in this video, you can see that we will be studying the classification of soil microorganisms. Firstly, we will study the soil classification of soil organisms, and then we will be focused on the soil microorganisms classification. And when we will be studying this, there are various kind of classifications that are important to study. The very first one is classification based on the ecology, then on the biochemical activities, then on the oxygen requirement, based on the temperature requirement, based on the mode of nutrition, based on the salt concentration. These are some classifications that are important and that can be asked in the various examinations. So hello everyone, welcome to the channel Agricultural Studies. My name is Biofertility, and without any further delay, let's get started. As I told you in previous video, we discussed the classification of soil organisms, and the soil organisms are mainly divided into two parts. The first one are animals and the second one is plants. The animals are termed as the fauna and the plants are termed as the flora. Fauna are micro and macro, subdivided into two parts, micro and macro. Micro means that are not visible with naked eyes, and macro are those which can be visible with our naked eyes. The macrofauna are earthworm, termites, and grubs, slugs, and snails, and centipedes, and millipedes, right? And the microfauna are protozoa, nematode, and rotipods. Whereas the microfauna, there are bacteria, actinomycetes, fungi, and algae. And in macroflora, there are plant roots and macroalgae. So this is something that we have already discussed in the previous video. So in this video, we will be discussing the classification of soil microorganisms. And when we discuss about the soil microorganism, that means they are not visible with the naked eyes, right? So there are various classifications which are based on the ecology, based on the biological activities. The first one, as you can see, the ecology classification based on ecology. There are microorganisms are classified as autochthonous, zygmogenous, opportunistic, and allochthonous. These terms are important, we should know about them. And other things like based on the biochemical activities, there are cellulose degraders, lignin degraders, ammonia oxidizers, nitrate and nitrite reducers, sulfur oxidizers, and sulfate reducer. And then there is temperature requirements. The temperature requirement, there are psychrophils and mesophils, thermophils, and you may know these terms also: aerobic, anaerobic, facultative aerobics, and microaerophils. So we will be discussing this from now. So the very first thing we should know about this is autochthonous classification based on ecology, and then we are studying autochthonous. What does the autochthonous means? Autochthonous means the indigenous. So microorganisms that are indigenous to the soil, that are already present in the soil, they remain in the soil, are termed as the autochthonous. So as you learned that autochthonous means a indigenous species of microorganism. So these are part of soil microbial community capable of utilizing humic substances. So this line is telling that these microorganisms eat the humic substance, or we can say insoluble material, especially the insoluble material. It is important. They eat insoluble material. They exhibit slow state of activity but are continuously growing. They exhibit slow state of activity but are continuously growing. They remain in the soil. They are indigenous to the soil and they grow slowly. Examples of these species are ascomycetes, actinomycetes, basidiomycetes, fungi, and some gram-negative bacteria. So when we will be studying the autochthonous, we will learn the autochthonous. It is starting from A. We will be learning two main important examples like actinomycetes and ascomycetes, which are of course starting with the A. So we can just remember this with a mnemonic that both are starting with the A, which are categorized in autochthonous. I think you may got the idea what are the autochthonous. And now when we discuss the zygmogenous or opportunistic, That means they are organisms which can utilize only simpler form. Zygogenous are also bacteria which are normally present in the soil, but the speciality is that they only can consume the simpler form of organic matter. So there was told that insoluble material, but when we discuss the zygogenous, they eat only simpler, often soluble substrate, right? They exhibit higher levels of activity and rapid growth on easily utilizable substrate that are available in the form of fresh plant litter. When there is a fresh plant litter, they can easily degrade them, decompose them, animal droppings and fresh carcasses. Then there is important line as intermediate activity or and inactive resting stage are characteristic of such zygogenous organisms. So we should know the two main stages, that intermediate activity and second one thing is inactive resting stages. These are some characteristics of zygogenous. Example, then we should know the examples. So there is PP balm. I learned this as PP balm. What is PP balm? PP balm can be remembered in the zygogenous. What is PP balm? PP balm is Pseudomonas, Penicillium, and then Bacillus and Aspergillus and Mucor. I have learned this. You can also learn this without any mnemonic and with other creative or better mnemonic also. So let's move to the next point, which is allochthonous. It is mentioned here as a note point that, as I told you, the autochthonous are the indigenous soil organisms. So the allochthonous means they are opposite to the autochthonous. Now, opposite means they are the foreigners. They are not generally present in the soil, but they are the foreigners. So when we say the foreigners means that, for example, pathogens of human, animal, and plants. When any bacteria, virus, or something which is a pathogen, means these pathogen needs the host as a human, host as an animal, host as a plants. But when they don't get these hosts, they remains in the soil for a little or a small period of time. That means they are temporary dwellers to the soil. I have also written this here. They are temporary dwellers of the soil. Okay, they are not permanent to the soil. If they are permanent, they would have called the autochthonous, right? So we are discussing for pathogens of human, animal, and plants that do not find suitable condition in the suitable condition in the environment. It should be in the environment to survive, get deposited in the soil, and then don't get any host around them. They deposit in the soil from various sources and are temporary dwellers of the soil. They perish soon if a suitable host is not within their reach. What is the meaning of perish? Perish means they get die. They will decompose. So they will die soon if there is not suitable host around them. They will soon get what? They will die. So in other words, these organisms which cannot survive in their vegetative stage in the soil, and those which do not have any adaptations to tide over the unfavorable environment are termed as the allochthonous. I think you may have got that what is allochthonous. Okay, what are allochthonous? So in next slide we are discussing the classification based on the biochemical activity of microorganism. So biochemical activity means if they are decomposing the cellulose, they will be called cellulose degraders. I won't be repeating this because I know you got this, right? The cellulose dwellers, lignin dwellers, pectin dwellers. So we will be learning these names as cellulose degrader are of course cellulomonas. The term also explains that the cellulomonas will be decomposing the cellulose. Bacillus are also example of this. Lignin dwellers, there are agaricus and coprinus, and pectin dwellers, there are Erwinia and Clostridens. Erwinia and Clostridens are example of pectin degrader. When we discuss the ammonia oxidizers, you may know the nitrogen cycle. I have also attached this here. Nitrogen cycle, you know that the nitrogen is firstly fixed as the ammonia form, right? It is known as ammonification, right? So the ammonification, or we can say the hydrogen is added to the nitrogen, right? So after the ammonia have made, then we there are some bacteria which converts this ammonia into the nitrate form and then the nitrite form, right? Nitrate form NO3, NO2 minus, and then the nitrate form, that is NO3 minus, right? You know that. So the bacteria, you may also have learned these name of these bacteria, like the conversion of ammonia to the nitrite will be done by the nitrosomonas, and then nitrite will be converted into nitrate by nitrobacter. So the nitrobacter and nitrosomonas we have already discussed. You may have learned these things in your previous classes. So nitrosomonas bacillus bacteria are the ammonia oxidizers, nitrate reducer. Why we are saying reducer? Because they will be get reduced to again the nitrogen also. Now again I have to make you see this, that the nitrate will be converted into again nitrogen when there are working of nitrification bacteria. So we can see that the denitrification bacteria are pseudomonas and acromobacter. Acromobacter and pseudomonas, you should have learned this two names. These are important. Okay, so next are sulfate oxidizers, thiobacillus species. Whenever there is word thio means they are indicating us to the sulfur, right? So sulfur oxidizers are thiobacillus. Then sulfur reducers are desulfovibro. Nitrogen fixers are symbiotic. Symbiotic relation means and they are two organisms are benefiting each other, right? So symbiotic relations are between the leguminous crops roots and the bacteria name is rhizobium. You may know that, right? Next is asymbiotic relation, have azotobacter and klebsiella. These are two important names, and the associative relationship can be seen in the azospirillum. So it is very important. So we have discussed the classification based on the biogeochemical activity, or we can say just biochemical activities of microorganisms, right? Next is classification of free living nitrogen fixers. When we study these topics in the books, we get confused a lot because there are various examples, and some are nitrogen fixers, there are aerobics, some are heterotrophs, and some are phototrophs. So we should have a knowledge as in a table, so we can have a clarity about these examples, like azotobacter, Wezerinkia, and Druxia. These are aerobic. Aerobic means they need oxygen, right? They are aerobic, but also they are heterotrophs. Heterotrophs means they want to consume oxygen, but they are dependent on other products also, like for the energy source, right? So we will be discussing these definitions of also in this video, heterotrophs and phototrophs also. So you should learn these all examples if you are preparing for competitive exams at higher levels, like for the PhD or for the SRF examination. You should also learn these examples. I won't be speaking these to reduce the time limit of this video, time duration of this video, but you can read them and you can learn them for their competitive exams, for the SRF exam or the higher examinations for the PhD entrance and all. Then there is a classification based on the oxygen requirements. You know that the obligate aerobes, they are totally dependent on what? They are totally dependent on oxygen, right? They don't have any other option, so cannot survive without it. They cannot survive without oxygen, right? And those which can survive without oxygen also can be known as the facultative, right? There will be word facultative aerobes. So how they facultatively can grow without oxygen? So there are because they adapt to the grow under anaerobic conditions, as they can depend also on the nitrate, sulfate, and carbon dioxide. Okay, so they are called the facultative aerobes. And there is important 9. An example of such organisms of great and agricultural significance in the soil is denitrifying bacteria. Denitrifying bacteria are facultative anaerobes. This is a very important line, you should remember this. And there is the organism which grow in the absence of oxygen are called the obligate anaerobes. Obligate, they are totally dependent on absence of oxygen. There should not be any oxygen. So these are also known as the aerophobes. They have phobia about the aero. Aero means the oxygen. So they are aerophobes, like the Clostridia and Methanobacteria. Methanobacteria, or we can say methanogen bacteria, right? You may have idea about this. So next is aerophils. There was also a term in the classification chart I showed that I showed you before. Yeah, this aerobic, anaerobic, facultative aerobics, and microaerophils, right? So microaerophils, what are the microaerophils? That those microorganisms which needs less, yeah, reduced level of oxygen. They need oxygen but and reduced level, right? So we can learn the concentration at 0.2 atm. Okay, it's an important definition that can be asked in any examination, right? This data is important, 0.2 atm, right? And the example is azospirillum species, something azospirillum. Okay, next we can see the classification based on the temperature, right? Okay, this is very important. Like bacteria or microorganisms that can survive below the 10 degrees Celsius are known as the microbes, which can grow in between 20 and 30 degree temperature, 30 to 20 to 35 degrees Celsius temperature, can be known as the mesophils. Meso means medium, like medium raised. So the mesophils, these are highly dependent, dominated. These are highly dominant and numerous in most of the cultivated soil of India. It's a very important line. You should know that in Indian soil, the dominant bacteria are mesophils. Then dominant microorganisms are mesophils. Okay. Then temperature higher than 45 degrees Celsius dependent on development of thermophils. Thermophils, or you may remember the term like Thermus aquaticus. Thermus aquaticus was an important example of thermophils, right? Thermus aquaticus bacteria can be seen in a very hot water vents and all, right? So commonly encountered in the post pits. So this is was something classification based on the temperature. I have also attached a table related to this. You can also read this. This is also very important. The temperature ranges are mentioned. The minimum and maximum temperature range is also mentioned. So you should also know these topics. So if the questions are asked in depth, they can be asked, they can come from this table also. Okay, you can see the extreme or hyperthermophils. There are, after thermophils there are extreme hyperthermophils, and I think I was telling about the archaea inhabitants of hydrothermal vents. I was telling the term Thermus aquaticus. I think it's the extreme hyperthermophils. Okay, so this is the classification based on the temperature, and then there is a classification based on the mode of nutrition. So you already know that the organisms are divided into heterotrophs and autotrophs. We have studied in various classes. So the heterotrophs are which are dependent on any other source of nutrition, right? Like which is dependent on any other substance for their energy source. But when we see the autotrophs are capable of making food by themselves, right? So the heterotrophs derive energy by oxidation of organic compounds. They oxidize organic compounds, or they consume compounds to take energy. But while the autotrophs derive their energy from carbon dioxide. Carbon dioxide, why? Because they have chlorophyll. They add carbon dioxide and H2O to form the glucose, and or we can say energy source. So we can or energy products for themselves, right? So this is something that we should know about the heterotrophs and autotrophs. The group of autotrophs is further subdivided into chemotrophs, and then there is the autotrophs. Chemo means they are dependent on something chemical. Chemical means they are dependent on inorganic substances. So there is simple inorganic compounds, so energy from oxidation of simple organic compounds. Then there is autotrophs which derive energy from sunlight. Okay, sunlight means, you know that what is the sunlight means. Okay, well, this is the very first video when I'm using English, when I'm trying to speak in English. I haven't uploaded these topics. I haven't made any video in English language, but it's my very first try, and I request you to just tolerate me for a while more. And I think after 2 to 3 videos I will be frequent to the English, and you won't get too much disturbed with the language issue and all. Okay, so there are students who told me that you should make videos in English also, because there are also students who don't get the Hindi language. I hope there will be also subtitles of this video available, but let's see what happened. And we should move to the next topic, that is, since most microorganisms require reformed organic carbon source for growth, carbon requirement is used to classify organisms. Microorganisms are classified on based on carbon requirements, right, as we discussed above. Then there is a term oligotrophy. This is an important term for the examination purpose. What is oligotrophy? So oligotrophy means which is used to describe organisms that can live in the environment with very low nutrient concentration, right? So what is about the higher concentration? When there is a higher concentration of carbon, very low concentration, then will be saying them as oligotrophic. So with a very low nutrient concentration is important within the soil system, which is often nutrient limited environment. When there is a nutrient or carbon limited environment, it will be called oligotrophic environment. Okay, when will be, so here the oligotrophic term is really important. You can see this term in the ecological studies also, and I know that it is an important term, but I am unable to add any example to here because I'm not properly getting the example of oligotrophic, but I know that it is an important term. You should research about this by yourself. Next is capiotrophic bacteria. On the other hand, they require higher concentration of carbon in the environment. Okay, so next is mode of nutrition again, but in a tabulated form, which can be helpful to you, like photolithotroph. Litho, when there is a term litho, that it means they reduced minerals. Okay, energy sources obtained from the light, because they are mentioned photo, and when because of there is mentioned as litho, it should be known that electron sources reduced minerals. And then is the photoorganotrophs have electron source. Organo, when there is an organo, means that organic carbon compound is the electron source. And when there is a photo aqua, when there is aqua, that means there will be of course water. Chemo means they and require energy from chemical oxidation, and litho means reduced mineral, right? And the chemo means should be there the energy from the chemical oxidation, but the electron source should be the organic carbon compounds. Okay, so this is what something you should have learned for the competitive exams also. Carbon source, these are the carbon source, which is of course mostly the carbon dioxide, but there is in chemoorganotroph there are organics. So this is something that is important for the competitive exams. Next is classification based on the salt concentration. There is a term halophiles. Means halo, you may already know that the halo means halo refers to the salt concentration. So the microorganism that requires some NaCl for growth. NaCl means a rock salt. So required is known as the halophiles. Based on the growth response to the salt concentration, they are classified as mild, moderate, and extreme. The mild are those which can grow in between 1 to 6% of concentration of NaCl. Moderate are those which can tolerate 6 to 15, and the extreme are those which can survive at 15 to 30% of salt concentration. Okay, then bacteria that are able to grow at a moderate salt concentration are halotolerant. So it is an important term, that I have italic given the italic font. No? Next is osmophils. Osmophils and xerophils can also be asked in any competitive exam. Osmophils are those which are able to live in the environment higher in the sugar content. Right? When above we are discussing the salt, so we should know the term related to the sugar also, which is osmophils. Then there is a term zero. The zero is related to the dry conditions. Okay, so this is what we should know about the classification, which is based on the salt concentration. Okay, so this is what I wanted to share with you as the classification of soil organism and classification of soil microorganisms, I should say. In this next video, we will be discussing the bacteria, or I can say we will be focused on the soil biota. Okay, I show you this. I have already shown you this we are discussing the subtopics as the soil biology. We discuss the history and important scientists and important classifications in this video. So now we will be discussing the soil biota. Soil biota is not a small topic, it is a very vast topic, as we have to discuss the bacteria first, and then there will be algae and all the organisms that are important with the point of view of examinations and of course for our knowledge. So we'll be discussing all these topics in upcoming videos. Till then, I would like to request you to like this video and share this video with your friends, and I hope this video will be helpful to you. Okay, so stay tuned to the channel. Bye bye, take care.